I'm a PPT artist actually.
I thought you were painting with
Yeah, we are live.
with data, you know.
Yeah.
Okay, we are live. Okay.
Okay. So let me see the participants. Okay. Okay, let's see. Okay, now. Okay. So let everyone enter actually. So uh, until we reach somewhere 75 or so.
Sure, Dr. Kumar.
Yeah. So I'm just preparing initial talk and okay. Professor Asif Khan is joined just now. So we have a few more minutes. Welcome, Professor Asif Khan. Okay, so yeah, so let's start. So basically, uh, Professor Dr. Muhammad Asif Khan is along with us today, and we are extremely sorry for the time confusion and time zone, due to time zone difference. We could not make it on the 4th. His talk was scheduled, and so without uh, a keynote speaker and a guest talk, any conference will not successful. So we had decided, requested him for providing us another date, and he happily agreed. for today's date. So with this background, we are starting. So Professor Asif Khan is having a joint appointment in Perdana University in Malaysia alongside Benjamin Lem Valley University in Istanbul, in Turkey. So so it's a double kind of double duties he has, and he's also president of EPO Bionet. So basically, he's a extremely busy person, uh, did his PhD from Singapore and worked in John Hopkins, and then John Hopkins Perdana joint initiative. He was also faculty in that initiative, with his strong background and strong bioinformatics and data science background. He will present today about viral diversity using a bioinformatics approach. Sir, stage is yours. Please uh, go ahead.
Thank you, Abhishek. Can you hear me?
We can hear you.
Okay, great. And you can see me. Hello, everyone. Okay, thank you for the kind introduction. It's actually Bez Mialim Wakaf University, so it's fine. Thank you, Abhishek, for giving me the opportunity to present today. And I apologize that things didn't work out the other day, but I thank you for extending the conference for today. Without further ado, I'll get started. So the topic today is analyzing viral diversity, as you can read. So if you actually break down the title, there will be probably two main keywords, or three keywords. Bioinformatics is definitely one of them. Viral diversity, which can be further broken down into viruses and diversity. So these are the three key areas that we'll be looking at today, and let's get started. I just wanted to make a note about the conference. I actually like the name of the conference. It's the first, so congratulations to Abhishek and the team for pulling this off. I really saw amazing number of speakers and a lot of participation going on. I managed to stay on for a little while the last time I was supposed to give the talk. And I wanted to highlight the title, which says data science in biology. Of course, to me, that essentially means bioinformatics, but I like the name because it's highlighting something that's going on today, that's happening around the world. I know data science is really big, and I like that this conference is trying to highlight that aspect for biology. When we talk about bioinformatics, sometimes that's lost. So this is kind of a reminder. So congratulations to the team for doing this, and hopefully there'll be many more such conferences in the future. Okay, so I want to start off actually by recapping. When we talk about viruses, one cannot escape from thinking about coronavirus. That's really happening right now. It's affecting all of us. It has changed our lifestyle. In fact, the way the conferences are organized, we are doing this online today because of coronavirus. Otherwise, probably we would be in Bangalore, face to face. So there was a talk I gave some time back, and while preparing for this, a little, short time that I had in preparing, so I saw there were one or two slides that I liked from that talk, and I just wanted to quickly recap. I'm not going to go through the whole thing. So this is actually from New York Times. This is actually some sort of recap, a profile of the coronavirus at 6 months. This was published when it just crossed the 6-month timeline. And so I highly recommend going through this. It's a very well written. It's very good for the public, for the masses, to be familiarizing themselves with what actually we are dealing with. So the ones in yellow is what I want to talk about very quickly. Background. So a virus at heart is information. sometimes don't think about a virus as just a packet of information, a packet of data that benefits from being shared. So I really like the way they actually put it up there. And this is gonna be something that I'm gonna highlight later as the definition of bioinformatics. And so the next point is trillions of virus species. So there are a lot of virus species, but only like 100,000, several 100,000 are known. So from that trillion, we know very little. And then fewer than 7,000 actually have names. And only about 250 are known to be medically important in terms of infecting humans. So it's a very, very tiny, small number. Luckily, it's such a small number. Otherwise, it would be probably very difficult for us. And every pathogen evolves along a path between impact and stealth. So I think this is rather obvious. So it's like a hidden enemy, or enemy in plain sight. So the other part was. Yeah, it's a mere wisp of data, just mere wisp of data, a small packet of data that has grounded the world and has prompted a collaborative response, unlike we have seen anything before. I remember flights were banned. I think in many places they're still banned. Only recently flights have started. So we literally grounded the world, planet Earth. All right, so some other things, perplexing array of faces. There's just so many things that we are still learning about this disease. You probably can recall back, we thought of this, and then now it's this, and then so many new things that are cropping up. We thought it would not be affecting kids, and now we see some new symptoms that kids are developing. And it seems like our communication is its transmission. This is also, I found quite fascinating the way they've highlighted. So us talking would be transmitting. I think there was a video about Japanese researchers trying to see even when we talk casually, transmission could be going on. So our communication, and that's why we wear masks. I have my masks, and I'm in a room, so I don't need to wear right now. We have our masks when we go out. It's become norm now to wear masks. The virus has no trouble finding us, but we are struggling to find it. So to know the virus is to know ourselves in reflection. With so many proteins in its toolkit, the virus has many ways to counter our immune system. And how often the greatest damage is not inflicted by the virus, but by the body's immune system. So sometimes, we have an like cytokine storm, you may have heard of it, over-response, immunopathogenesis. So the damage may be not by the virus, but really our immune system in trying to kill the virus or get rid of the virus, ends up damaging, there's some damages, collateral damage, you might call it. So the other interesting thing is, 2 mutations per month, which is actually less than half the error rate of the flu. So of course, less, so there's maybe better chance of vaccine. For flu, we have this annual vaccine. But at the same time, because of the transmission and because of the rate it's spreading, even though it might be less than flu, because it's just the transmission is so facilitated, it might be harder. Well, there are a lot of efforts, so hopefully we'll get a vaccine quickly. But we may have to produce vaccines regularly if the mutations, if the spread doesn't get curtailed. So the next point, I think I've repeated. So it's mechanical, unreflecting, consistent on message. This is on the virus part. So the purest near living expression of data management on Earth, which is what typically viruses are. And us humans are exactly the opposite and complex. So two opposing forces, and we are struggling, and hopefully we'll beat this in time to come. All right, so COVID-19 is not the only pandemic. Of the many pandemics of viruses, many pandemics in the history of mankind, a large number of them have been viruses. So I think the plague, of course, plague has been a big one, but you have smallpox, you have yellow fever, for which we now have a vaccine, quite effective. Spanish flu, the big one there, this is flu 40 to 50 million, Russian flu, of course, HIV is still around. Then we have Asian flu, Hong Kong flu, a lot of flu. And of course, coronaviruses have been around too. We have SARS and MERS. Ebola is there, swine flu, COVID. And something to note, although the size of the ball there is rather small, that's like June 17, do not know exactly what that number, from when that number is, but I think the number is much bigger now, definitely much bigger. I think in terms of infections, 26 million, if I'm not wrong. So yeah, the ball would be probably quite big now, maybe competing Spanish flu, coming close to that, or HIV for that matter. All right, so this is not something new. This is something mankind has been dealing with, and hopefully this will pass too. This is something I borrowed from one of my students, James Among, Dr. James Among. Now he just finished his PhD. So this is just a picture of the most lethal viruses. So some of these you might recognize. SARS is of course there. They may not be lethal in terms of infection, but given the spread, a number of factors, they have been labeled to be the most lethal. So this is just something to keep in mind. Some Zika, for example, we had a bit of Zika scare some time back. All right. All right, so that's a very quick overview of viruses. Now, the second keyword is bioinformatics. Now, everybody, I think, is more or less familiar. So very quickly, just to recap, bioinformatics has been a lot in the news for a long time, and more so now, and especially with data science. So bioinformatics briefly is a bedrock of current and future biotechnology. There's a very nice article that you can refer to. It's a key driver of life science that's what I like to think of. It's backbone of life science. So essentially, this conference I was talking about, the title of this conference is about data science and biology, or life science for that matter. So essentially, this conference is about bioinformatics. Hopefully, Abhishek would not disagree. And data science is lifeline for Industrial Revolution 4.0. So you can really see how important is this area. And I hope more of you, the younger ones out there, would take up this field and make some new discoveries, bring innovations. All right, so definitions. What most people know about the definition of bioinformatics is the development of application of techniques from computer science, blah, 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 to address biological problems. This is what we know of. I prefer the more philosophical definition. It's one of the many out there, is the study of the information content and information flow in biological systems and processes. And if you remember the first slide, first two slides about coronavirus, COVID-19, the virus itself is just a mere wisp of data. It's packet of information, and bioinformatics is perfect to study that packet of information, because it's the study of the information content and information flow. So it's very well suited for this problem, and that's what we're going to talk about. All right, of course, to study bioinformatics, to do bioinformatics research, you need data. So what kind of data? Of course, viral sequence data. So very quickly, we do have a lot of data available. In fact, if you go to NCBI, there's just a lot of data, and the data is growing quite rapidly. This is from one of my students, Chong Li Chuin. So we actually did a bit of a simple thing, tracking the data growth. So in blue, you have the nucleotide, in red, you have the protein. And so for over a period of time, you can see the proteins, from 4.9 million to 5.8 million for over a period of a year, and that's 18% growth approximately. And for nucleotides, about 10% growth. All right, so data is growing rapidly. I think it's more than 5.8 million right now. So James actually studied this whole thing for one other research project. So data is available, abundance of it. And one of the reasons why I love studying viruses, because data is abundant, it's publicly available. And of course, the genome size is smaller, so it makes it easier to do a lot of fun and interesting research work. Now, just to quickly share for those who are interested in studying viruses, they just want to quickly run through the portals that you can actually avail to for data. Taxonomy browser is one of my favorite, because it actually allows you to download data using the taxonomy lineage, which makes it much easier. Unlike doing a keyword search, you go to NCBI protein database, you do keyword search, there are a lot of false positives out there. So this is much better. It's like using ontology. They're using ontology to organize it. So I hope people would actually use this a lot more. So you click on viruses, you can see, so I think this is quite a recent one. So 6.8 million. 6.8 million has grown a lot more, but it must be much larger now. This is not the latest. And for nucleotides, so you can actually, of course, you can't download this through the web right away. You have to use one of the NCBI NCBI APIs, and I think they use e-utilities. Yeah, API. You can download this because it's big. It's going to take some time, maybe 2 months. One of my students, I think two of my students took about 2 months to download the data. So yeah, you can download them. Smaller sizes of these you can download from the web itself. And of course, there's an FTP version. So what I like about this is, for example, here, the lineage, you can see down here. So down here, the lineage, this is, sorry, the circle is a mess. So you could click on any one of these and you could download data at any one of those taxonomic lineage levels. All right, so that's the best part. Moving on, so yeah, this is what I mean. You could actually go by the lineage, click on any one of these. For example, SARS is part of this. You click, you can, then SARS will be one of the subsets, and you can click on that and you get all the data. Besides the primary sources like NCBI, you know, there's some sort of specialist sources now focusing on certain aspects of organisms. For example, NCBI virus is just focusing on virus. So they also extract a lot of this data from NCBI protein databases and actually organize them in a much better way. I like to call them a specialist databases. So you could also get for certain viruses, I guess, important viruses from here, but I don't think they have everything. So you may have to go to the other one if you want more. For example, the other specialist resources, VIPR, they have their own nice organization of data, and they also provide, for example, coronaviruses and also several other viruses. So you could also check out this one. GIS8 has been actually very interesting. It's a very interesting example. I think they've been focusing on flu for a while, and recently they actually have also facilitated created a data repository for coronaviruses, and they're doing a very good job. One of the nice things about this one is that this is very interesting. Like, for example, for flu, they actually have kind of surpassed the importance of the primary databases. So sometimes, like for flu and corona, you might find more data here than NCBI virus database. NCBI protein database for that matter. And there are reasons why people actually prefer to dump the data here. I don't have time to explain all that, but you definitely want to explore this. I always cite this example. We submitted a paper and then the reviewers came back and said, why didn't you look at this one? And then we thought, well, how different could it be? And when we opened and we checked, actually, there was so much more data here than what we thought would be the most complete database out there, which was FluDB for a flu paper. And so we had to kind of redo. So do check this out. You don't want to be redoing your work, later on. But I don't think they cover a lot of organisms. So far, what I know is flu and coronaviruses. So if you're studying flu, you're studying coronavirus, you definitely want to look at this one. And they also provide very nice tools for all kinds of analysis. And of course, there's some country-wide, country-specific tools also out there. So, for example, China National Center for Bioinformation actually provides provides data for coronaviruses, and I think they do both local and global. So you may also want to check out whether there's some sort of a specific resource, a country-specific resource that does both. So don't miss out. It's good to do a comprehensive search before you proceed. Now, coming back, okay, to the diversity part. So why viral diversity? Why is such a big deal? Well, viral diversity, they mutate rather rapidly, or the rate of change is much higher than eukaryotes for sure. Even, they outcompete the bacteria. And RNA viruses in particular are among the highest ones up there. So for example, you have the DNA viruses, single-stranded, double-stranded, but the RNA viruses are among the highest that change. So this term quasi-species, roughly what it means is, if one person is infected by one virus, after some time, that one person would have some sort of a spectrum of variants within that one individual. So this is, akin to a quasi-species. So you have, you start with one and you end up dealing with a number of variants of that particular parent infecting virus after some time. So that's how bad it can be from, within just one individual. In fact, for HIV, there was a study where you could have 2-3 variants in your body, circulating at the same time or infecting you at the same time. All right, so coming back to the diversity, just wanted to highlight that there are actually 3 ways you can think of diversity: genetic diversity, protein diversity, and antigenic diversity. Genetic diversity, I guess everybody knows, these are the changes at the nucleotide level, right? So you have one DNA, then the second, let's say, a variant comes in and there are changes at the DNA. So that's your genetic diversity. Protein diversity, is a little different, because not all genetic changes will impact at the protein level. You know, you have synonymous mutations, non-synonymous mutations. So it's the non-synonymous mutations that actually affect the protein, level. So, so they might. So a lot of these synonymous mutations wouldn't actually impact the protein. It's the non one. So you may have something at the genetic that has no impact at the protein level. So this protein diversity is focusing only at the level where, you know, amino acid changes are actually happening. Now, antigenic diversity is also a little different. It's a smaller subset of protein diversity, and protein diversity is a smaller subset of genetic diversity. So what do we mean by antigenic diversity? This is diversity that's relevant to the immune system. So what do we mean by that? So if you take a protein sequence and identify the epitopes that are in this protein, epitopes are basically parts of the virus that are recognized by the immune system. Now, if there's a new variant, a new strain, and there are mutations, but some of those mutations may not be at the part where the epitopes are, and some mutations may be at the part where the epitopes are. So some mutations will impact the epitope, some mutations will not impact the epitope. And sometimes some mutations may impact the epitope. So in the sense that there's a change, but from the recognition point of view by the T cells, for example, there's no difference, because the T cells can also tolerate some sort of variation. So whether even though there's a change, it doesn't impact recognition. So it's a whole different level. It's a whole different thing that needs to be analyzed in its own way. And depending on what, for example, HLA you're studying, or what, T cells or B cells. So antigenic diversity, it's a little different. And so sometimes you can have protein changes that don't impact it. Sometimes you have protein changes, there's a change, but it may not be in the epitope region. There is a change, it may be in the epitope region, but it may still not impact the epitope. It may not abrogate the epitope itself, or there might be a change and then it does abrogate. So there's that other aspect that needs to be considered. All right, and of course, these antigenic changes are bad. Bad in the sense that they can result in immune escape. You can have a variant or new strain that escapes recognition from the immune system. You can have original antigenic sin. This is quite well known. Well studied for dengue, for example. You recognize one serotype of dengue, you get infected by the second serotype of dengue, but they look very similar, but yet they're very different. So instead of triggering naive cells, naive T cells, you call them memory T cells, and you attack the pathogen, but the response is not going to be effective because they're actually quite different. All right, so that's termed as original antigenic sin, where instead of mounting a new response, you mount an old response, and that response doesn't seem to be quite effective. And there have been, this has been linked to something called immunopathogenesis. So immune system is attacking, but it's not effective. So you also end up damaging your own body cells. So original antigenic sin is something else that could happen. Of course, this also makes it much harder for us to design vaccine. This is something I mentioned earlier. So, for example, in flu, there's a lot of change, and we have actually annual vaccine formulation for flu. You probably have walked to a clinic and say, "Oh, we have this year's summer vaccine available for flu." You may have seen one of those adverts, not adverts, but basically info. Labels. All right, so now I just wanted to highlight that antigenic changes are more punctuated than genetic changes. What does that mean? So let's think about this. Okay, so let's say this is a protein, and we have this position 9. Okay, and if we were to generate, epitopes. Let's say they are epitopes here. So epitopes are typically, in the case of T cells, they're typically 9 amino acids. So for example, how many epitopes can you potentially have? So potentially you basically do overlapping 9mers. So 1 to 9, maybe one potential epitope, 2 to 10, 3 to 11, and so on and so forth. So you create all these potential k-mers that could, all these k-mers that could be potential epitopes. So you have to test them out. But let's just hypothetically, say that all of these are epitopes. All of these are epitopes. So if there is there's a new strain, and at position 9 instead of W, let's say there is something else, phenylalanine, for example, then it's gonna affect all these 9 epitopes. It's gonna affect epitope 1, 1 to 9, 2 to 10, 3 to 11. It's gonna affect all of this. So one mutation, you know, in the antigenic sense, one mutation can actually impact can span, the impact of one mutation can span a region of 17 amino acids, right? So it's asking us to think in a little different way. It's not the same as just saying, okay, there's a mutation here and that's it. But from the immune perspective, that single mutation is not just single mutation. It can actually change 9 potential epitopes. So we got to be wary of these 9 potential epitopes. You know, not just be concerned of the first epitope, but all of these 9 epitopes and how they may actually impact how the recognition may be affected. You know, so we got to study that. We got to look into all of that, and why the recognition would be different. Well, for the first one you have 1 to 9, but for the second one you have something extra, which is number 10. For the last one you have a lot of different. The only thing that's identical is just this 9th position. Everything else is different. So yes, the response could be different. And you know, impacted by this change. You could look more into this. You could read this Ramenzi et al.'s paper, but that paper only states that, you know, why I chose the size 9. Size 9 is the typical length for MHC class 1 epitopes, T cell epitopes, and it's actually the core length for MHC class 2 T cell epitopes. I am focusing mostly on T cell response, T cell immune response, cellular immune response, because this has been very well studied. Antibody is a whole different matter, a little because you have conformational epitopes. Okay, so I hope I have explained, you know, antigenetic, protein, and antigenic. So the focus of my talk is going to be actually more on antigenic diversity, and I'll explain more a little later in terms of the methodology. So, but also we can loosely, you know, think of studying viral diversity in 2 different ways. This is actually a slide borrowed from my student Chong Li Chuen. Thank you, Chong. So, so think of, you know, well, you're looking at diversity, and you could do it in 2 different ways. Well, one is alignment dependent, another one is alignment independent. Now, for a lot of people, you know, they might say, why do you want to do alignment independent? It wouldn't make sense to us to do alignment independent. Of course, I think most of you would be familiar with alignment dependent. You align, you look for conserve, non-conserve, and etc. My focus for today will be alignment dependent, not so much on alignment independent. But I want to just highlight that, you know, alignment independent is equally important because, for example, if you want to study all viruses and look at the diversity, it's going to be, I think it's going to be impossible. It is impossible because you can't align all these viruses. You wouldn't find homology. So alignment independent, it's an important area that needs to be looked into. You know, we have limitations of alignment dependent approach. So actually, Chong, I mentioned that student has done a lot of work. That's a separate talk, separate discussion. But I'm going to give you a brief an overview how the problem could be approached. So alignment independent, well, simply not using alignment some other way. And I think there are many different approaches. I've seen other people's work looking into this, but not so much into diversity. But I've seen phylogeny, people do phylogeny using alignment independent approach. I saw it, this talk by, can't remember the guy's name, but yeah, from India. It was a very good presentation. All right, so alignment independent approach. I just want to highlight this concept. So for example, we have this idea of a minimal set. We are working on a paper for this. Actually, there's some earlier publications you can refer to, actually very old publications of mine that you can refer to if you want to know more about this. So for example, we have 3 sequences and they're all unique. I mean, you see there's some difference, right? So the sequence 1, for example, is FFL. So the 3rd sequence has L here. And you have the first sequence has Q, which is different from the second one, and the second one has L here. So overall, they're all unique. They're all unique sequences. You can come up with a consensus and you can identify the variable residues. Now, if we do the same thing, generate potential epitopes from and use the 9 amino acid as a length, then you get this repertoire of 9 mers from each one of these. Now, let's analyze closely. So this set here is actually identical. These two sequences share exactly the same repertoire for that's in the blue box. And for these sequences, this set is exactly the same. And the red ones are exactly the same across the three of them. So you notice something. Actually, the middle sequence is somewhat antigenically redundant. You know, all the possible k-mers that, or the possible epitopes that can come out from this sequence are already covered by the first sequence and the third sequence. So what would be the minimal set of this? The minimal set of this could be 2 out of 3. So you have 3 sequences, but you actually need to study 2 sequences, and these 2 sequences would capture the antigenic repertoire of all the 3 of them. So that's the idea of a minimal set. And we take this idea further. There's a lot of work that has been done by Chong, and hopefully we'll publish that work and we can share with everybody, and you could read into that. So that's one. But coming back, we're going to be focusing on alignment dependent. So for alignment dependent approach, there are 2 papers, key papers that I want to focus on. This is our earlier work on HIV that we have done. It was published a while ago, 2013, and this is our most recent work on flu. We just published May of this year. I will be focusing more on the flu since it's the most recent one, but I'll draw some references to the HIV one. I'm just keeping a tab on time. Actually, so all the stuff that I'm going to present today, the results, actually we are trying to build a tool called WADA, and WADA hopefully will be able to automate the results. You're going to see this, so other people can do the same thing for any virus of their choice. So this is something we are working on. It's still at a prototype stage. Hopefully we'll get this out soon. If you need more information about this, do let us know. All right, so this is the paper that I'm going to talk about. This is just a quick overview. It's if you want to read more, this is the link, and it's published on in PeerJ. So this work is focusing on influenza. So I'm going to use H5N1 influenza as just an exemplar, just to demonstrate the work that we have done. So, of course, everybody knows influenza virus is a big health problem worldwide. You know, there's a regular flu you know, there's a regular flu season. The disease spectrum can be mild, can be severe. History of influenza pandemics, of course, the big one is the 1918 Spanish flu, and then you had several more. And there's a H5N1. There was a big scare of H5N1, you know, a few years ago, and it still remains to be one of those with potential for actually pandemic. There's another one, H7N9, that actually has higher potential for pandemic, but H5N1 is among one of them that is being watched out for. So this is something out there that could come back, you know, in a big way. So we got to be wary of that. So this is just a quick report. There were a number of deaths that have been reported, and the annual influenza vaccine does not cover H5N1, so we need to be concerned about. And so currently there is no commercially available vaccine against this virus. This is the genome of the virus. So it's a segmented virus. I think it's negative sense segmented virus. So you have like 8 segments, and each segment, some of them express one protein, some of them express multiple proteins. So they're like a polyprotein. Some of the segments behave like a polyprotein. Some of them are just independent. For example, dengue is another story, purely polyprotein. It has its own challenges of managing. But you can see, so it's not a big genome. And there are 8 segments, and then there are like actually in total 12 proteins that you have to deal with. So it's manageable, workable, but you have a lot of data. Flu has a lot of data besides HIV. HIV, of course, has a lot more, but flu has a lot of data. So the bigger problem here is dealing with that large quantity of data, especially for highly diverse proteins like HA, hemagglutinin, and neuraminidase. They are quite diverse, so aligning them is a challenge. Okay, so these are the proteins, and I don't have time to go into the functions of the protein. You can read into. So, and of course, these proteins, you know, they go through antigenic shift, antigenic drift. Antigenic drift is where your regular mutations, and these mutations are usually responsible for the epidemic, and it's antigenic shift. That's a major change. For example, you have reassortment or recombination, and you know, it creates a big change, and antigenic shift has been associated to be responsible for the pandemics. All right, so these are the two major forces of change, and there was actually a viral diversity analysis done by a colleague of ours, Haney et al., published a long time ago, and this was just a small part of a bigger study, and we wanted to just have a look at that as our starting point. So this is some sort of a reference for us, and she did some entropy plot. Entropy plot is a way to study diversity. So you can see the diversity for the avian and the human, so the avian population more like the reservoir and the human, so you can see. And but this is just a plot. It gives you a panoramic overview. We felt that there should be more that could be understood about the virus besides getting an overview. Of course, this is great, but we needed to be able to do more. So our goal was a great understanding of influenza proteome sequence diversity, the dynamics of mutation, or rather the change. Actually, the word mutation may not be appropriate because the change could be because of mutation, because of reassortment or recombination. So I think the better word is substitution, and that kind of encompasses all the three, because we are not really looking at specifically what's causing it, but we're just looking at the outcome of the forces, right? So I take it back. I think the word substitution is more appropriate, and of course to see how we can overcome the diversity. For vaccine design, I'm not going to focus so much on the vaccine design. That's a separate talk, but yeah, you'll get the idea. All right, so this is the methodology. Quite standard these days. You download the data, you went to the resources I mentioned, and you download the human and the avian. So if they are groups, you've got to, it's good to separate them rather than mix them and then study them together. It makes it much murkier. I think separation, you break and conquer. It's a much better approach, and then you'll also get to compare between the two. Gives you a better overview of what's going on, and then so you download and then you separate, or you download separately. I think it's the FluDB, for example, makes it easier to download separately. I think the other databases also allow you to do that, and you also then can study the host distribution, for example. Of course, for human it's easy. You can look at the countries where they're coming from. For the avian, on the other hand, the reservoir, you can look at what are the birds that actually contribute the sequences, and what are the countries they're coming from. So you can do that distribution and get a nice overview of what's happening. And then you group them by protein. So of the 12 proteins, so you have avian 12 proteins, human 12 proteins, and then you map the proteome sequence diversity. You can do the entropy plot like I showed earlier, and you can, and of course we'll get a much updated overview of that. And then you quantify the dynamics of sequence change. So the word change is much better here. And then you can do further analysis of that. And this is where we are designing an automated computational framework. I mentioned WADA. All right, so data collection, this one, we used FluDB, which is one of the good ones out there. So, for flu, like most people actually don't go to NCBI anymore. They either go to FluDB or GIS-8 EpiFlu. But you could, do all of them just to be sure that you've captured everything properly. So we collected the data, removed duplicates, multiple sequence alignment, do QC, multiple sequence alignments. You have to do eyeballing, make sure that the alignments are correct. Misalignments can happen, especially if you take short sequences and you do the corrections. So before alignment, and you can see there's a big difference after, well, I know this is just simply alignment, but yeah, editing, it's an important task, and then you better make sure that you have thoroughly checked the alignment before you proceed, otherwise there could be issues. So here's an example of the data. So you can do countrywide distribution, and you get to see where the data is coming from, and you can break down the numbers according to the proteins. So this is the breakdown. All right, so this is a quick overview. We had a total of 13,000 sequences, so not so much, not so many for H5N1, and most of them from avian, and human only about 1,800. And this, bias, it's a reflection that in human it's a recent, it's a recent case. So only now people are studying, but for avian they have been actually looking at this for a while. So we have more sequences for avian. Now, okay, so once you have the data, you have aligned them. Okay, then what do you do next? How do you study antigenic diversity? So actually the right thing to do, so this is just a picture for cellular immune response. You have your T cell, the T cell receptor, and you can have class 1, class 2, CD4 is class 2, CD8 is class 1, and you have your cell, antigen presenting cell, which will, the virus comes in, basically gets broken down to small peptides, binds to something called MHC, and then you have class 1 or class 2 MHC, and the MHCs basically go up to the surface of the cell, the T cell comes, binds, and recognizes, and the antigenic fragment is your epitope if it gets recognized. This actually binding peptide is, if it gets recognized, it's a T cell, it's a T cell epitope. And for class 1, it'll be typically 9. For class 2, it's much longer, but you have a core region of 9 with some flanking residues that strengthen the binding. All right, so let's say, you know, so we decided that we'll stick to 9 as a length to make it easier for us to do the analysis. So 9 was the window length that we decided to use. Okay, so for example, so how do you do the diversity? Then you take the alignment, you basically extract the windows of the overlapping windows of those 9 and then do diversity analysis on that. So for sorry, yeah, so okay, so yeah, you extract that 9 and then you do the analysis on that. Okay, so how do you do the analysis? We used Shannon's entropy. And instead of using for a single amino acid, we basically applied it for a window of 9 amino acids. But otherwise, the concepts are the same. We have written a lot about this. You can read our various papers. So PIX here is the probability of a particular nonomer. So 9mer is a nonomer at that position X, where you basically you're studying. For example, position 1 to 9, then the middle position 5 becomes the reference position, and you basically look at the diversity at that position. So probability of particular nonomers. So how many different nonomers are there at that position, and what is the probability of those nonomers at that position? NX just represents the number of peptides observed at that position. So for example, this is for dengue NS2A, this is the position, and 176 is just the middle position, and there's actually 64 sequences. So NX would be the number of different peptides you have. So 1, 2, 3, 4, 5, 6, 7. You have 7 different peptides or nonomers, and this is the frequency. So NX, the PIX, put it together, you get an entropy value, 2.40. It gives you an overview of the entropy at that particular region, and then you get to see what's going on. So entropy is affected by the number of unique peptides and the relative probabilities. And this is just to show that what is the maximum entropy you can have, 39. And what is the peak entropy we have seen so far in our study? Actually, for HIV, we have seen an entropy of 10. All right, so 10 is the, so 39 is the theoretical one. The one observed is 10. So that gives you an idea. You know, HIV is known to be quite plastic. In fact, it has regions that mutate a lot. So 10 is the highest that we have seen. This might be the highest out there, maybe not, but it's pretty close to the observable highest. And for example, West Nile virus is very conserved, is about 2. Dengue, the peak is about 4. For flu, we have seen about 6 to 8. So hopefully that gives you some perspective. So when you plot, if it's high, highly diverse, entropy is a measure of, entropy of, basically, I'm looking for a particular word. Disorder, yes, disorder. It's a measure of disorder. So the higher the value, the higher the disorder, therefore the higher the diversity. The lower the value, the lower the disorder, therefore conserved. So that's the correlation here. Actually, we have a tool, ABK Awana, that you could download and use it if you want to do your own entropy plots. This tool actually does so much more mutual information and other things. So you could check it out. There are blogs out there that are written. You can see some tutorials out there. So coming back to the results, Abhishek, I think am I okay still with time? Can you just give me a heads up of how much time I have? Or I've already run out of my time.
You can go, no problem, because you're the only one speaker. So you can, if you are
okay.
having some time,
Okay.
not issue.
All right, that's a privilege. Thank you. Thank you. Okay, so entropy analysis, this is the result. So for avian viruses, human viruses. So if we look at proteome-wide average entropy, it does look very similar to what Haney had earlier obtained. But of course, we have more data now, so we'll see more diversity. Quick statistics, proteome-wide average. So this, of course, there are peaks that you see that are reaching 6 and coming close to 8. But we're talking about proteome-wide average. You know, overall. So overall average is actually rather low, 0.8. To me personally, anything below 1 is quite conserved. We'll see what is the correlation between entropy and total variance. By the way, I forgot to mention total variance. Let me just quickly go back. Yeah, so there's another concept I want to highlight, which we had established actually earlier. The sequence that's going to be highly presented with highest probability, we call that index. And we use the term total variance, which is to describe everybody else. So basically 100% minus 39%, which is your index. That's the total, the incidence of the total variance. All right, so it's a very simple concept. You have all the peptides here, and you take the dominant one as the index, and everybody else is a variant to the index, and you get the frequency of that of all the variants. Okay, so coming back, yeah. So so in red here, what you see is total variance actually, and in black is the entropy. So when you have entropy high, we expect total variance to be quite high. When entropy is when entropy is 0, so you have basically one sequence that's 100% conserved, so your total variance will be 0, because that one sequence that 100% that is 100% conserved is index. So if index is 100%, your total variance is going to be 0, right? So you see some very low ones. You don't see anything red because then the variance are very low. So if the entropy is low, the variance is going to be low. If the entropy is high, the diversity goes up, the variance also go up, total variance percentage goes up. So you have this red and black lines here depicting both, depicting them. So anyway, coming back, so proteome-wide average entropy was actually rather low, 0.8, and the smallest entropy you can have is 0. So, and 0, as I said earlier, is 100% conserved. You have one peptide that's 100% conserved. So you can see it's very close to 0. And which means in general, H5N1 is actually highly conserved, which is a good news. And the total variance proteome-wide was 14%. Of course, this could be very high. In the case of HIV, this is very high. Of course, the average is not that, but there are pockets that can be very high. So on average, it's 14%. Right, so that tells you that you have your index on average would be 86%. So if index is 86%, then your total variance is going to be 14%. All right, so your index on average, most of these positions have one sequence that's around 80% in general. All right, so and what about the human viruses? The human viruses were total variance wise, they were roughly the same, and entropy was slightly lower. So overall, it's a good news that these viruses are not that diverse, and for vaccine, in terms of vaccine, there's a lot that could be explored here. And you do have a lot of pockets, for example, in the human, where the entropy is just very low. So instead of throwing a whole virus and not sure which part might get recognized, because you do have some quite diverse parts here, you could just use a different approach. Peptide vaccine approach or epitope vaccine approach, where the immune response could be targeted on these conserved regions, and that way we can avoid these diverse ones from being recognized. The diverse ones also are thought to be quite immunodominant, so we want to avoid them. You put all of them together, you throw a whole virus, the diverse ones actually like bullies, they will push away the conserved ones and would preferably bind to the MHC and be presented. And then when you have the next strain, it's going to be different. So your immune response will not be that effective. So you do have a lot of pockets of conserved region that you could focus on. All right. Of course, the individual protein average is going to be much lower. Entropy wise is going to be lower, but this is actually presenting the range. So you can see like PB1F2 has quite a high total variance. So that's one of the proteins that's quite diverse. Well, probably the diverse among them. Of course, HA and NA are also diverse. HA is for entry, NA is for exit. So these two proteins have been a hot area of research for vaccine design and drug design. Okay, so this gives us an overview, but this is it. This is a panoramic overview, very nice. This is what we asked earlier. Can we do more? Just for comparison purposes, we have the HIV data, and you can see for HIV, it's just a whole lot different. You see very few pockets of much lesser than H5N1. This is actually just one clade of HIV. clade B, and you can see, you can compare the diversity, and you can see that, for example, this position here, it's actually a beyond 8, so it's coming close to 9, and this is the highest we have seen. And so what's amazing about HIV, you really have these positions, some of these positions that just keep changing. It's like they're so plastic. There's no restraint in terms of function. It's just amazing how some of those positions can mutate so much. We'll talk more about that later. So this is just to correlate. Okay, we were just curious to see entropy versus total variance. What kind of relationship do we see? So when the entropy is closed, then total variance actually, it's very tightly regulated. But as entropy grows, as you know, entropy, the larger the entropy, more diversity, you do see a spectrum of total variance. So for example, 50% total variance, you can get this 50% total variance at an entropy of 1, at an entropy of 2, at an entropy of even 3, right? So 50% total variance can come from a range of, for a range of entropy values. Why is that so? Because actually it depends on the dynamics, the composition of the total variance. So this highlights that we need to look into the composition. We kind of made it very simple, binary, index total variance, but there's a lot going on inside the total variance that we needed to break down and have a look at. And so this highlights that something interesting is going on, and we need to understand that. Human viruses were somewhat similar, but yes, well, the trend was somewhat similar, but there's of course some differences between them too. So it's not exactly the same. So we were curious, and we wanted to dig deeper and analyze further. And the same thing we observed for, of course, HIV. But you can see the plot for HIV is a little different. It's more of an upward trend. It's a bit much tighter at the lower end, and it's actually much more tighter, much more less spread out than the flu. And has a slightly different trajectory also. So you can see that it's not the same. You might, okay, do all viruses look more or less the same? And actually not the case. So it's interesting to do these kind of plots for different viruses just to see what's going on. So we wanted to break it down and have a look. So the conclusion of this is that it's necessary to look at the composition of total variance. So we said, okay, let's come back and to our entropy calculations. And for example, you have this position, and you have the index. This is the big one, 16. So it's going to be the index, and you have these others. So we got to study these. So how do you study this? What other ways one could look at this? This is just a formula of how you calculate the entropy and the total variance. So you should know this by now. So let me give you an idea how we actually broke down this. So we actually came up with diversity motifs, and this is a definition of the diversity motifs. All right, so the biggest, for example, you have 20 sequences, and this one is highly represented. The dots mean identical, so 8 out of 20, so that's going to be the index. And then you have another one that's 5 out of 20, so that's going to be major variant. So it's the the second dominant sequence. But it's a variant to the dominant one index. So we call this this is one of the variants, and it's going to be the major variant. All right, so it's second most represented sequence. Then we had few others that we observe. For example, unique. They were only observed once. Okay, so this was very interesting. You only see them once and then they disappear. So we call them the unique ones. So 3 out of 20. Then everybody else, we lump them together as minor variants. So they are represented more than the unique, and they are less than the major. They are not as dominant as the major. So these middle groups would be the minor variants. And we also came up with another term, nonatypes. This is just to depict how many different variants do you really have. So of course, the major will be 1. The unique, in this case, produces 3. You can sometimes have more unique. So in this case, it's 3 plus 1 major, and then 2 in the minor. So 3, 4, basically 6. So you have 1, 2, 3, 4, 5, 6. So the term nonatype is to basically depict how many different distinct variants do we actually have among the total variants. So we broke down the problem into this. We broke down total variance into this, so we can actually look at the pattern for each one of those. All right, so this is a quick definition. I've already explained, so I'm going to move on. This is just a sample. So for example, at a position, so we ran through the whole proteome alignment, and we came up with something like this. So the position, the number of sequences, Hx is the entropy value at that position. This is the index sequence and the frequency of the index, and the total variance frequency. So it's simply 100% minus the index, and then you get 8% here. Then we got the frequency of the major, the minor, the unique, and the nonotype. So this is what we did. We got all the data, and then we started plotting. All right, so this is for avian and for human. The red line is just the index, and the black line is the total variance. So of course, index will be going down because this is a plot of motif incidence versus total variance. So your x-axis is total variance, and that's why the black line is going up, because total variance is going up. So when total variance goes up, it's an inverse relationship. Index will come down by definition. So you do see that, that's expected. But what is not expected is whether you actually have index that looks like a straight line. Actually, it's not a straight line. These are just dots, and it appears like straight line because all the, you actually see index of all different values. And if you see all the different values, you'll see a straight line, but you see some breaks here. There's some parts here that do not appear. There's a blank, there's some blank, and it gets more, so for I think 80% onwards, 80% total variance onwards, you actually have no positions. Actually they don't have any positions beyond 80% total variance. So we get to see what's going on. And what's this green one? The green one is the major variant. It actually has a pyramidal shape, and that's by definition. So the pyramid is not what we are interested in. What's interesting is, what part of the pyramid do they actually occupy? So for example, why is it a pyramid? Well, because major has to be below the index. So when your index is very high, your major could be higher, but unfortunately, well, by definition, it also has to be below total variance. So they're restricted. So if your index is, for example, 90%, your total variance is going to be 10% and 10%, and the major has to be below that because it's the dominant within that 10%. Okay, so this black line, the major has to be below that. But as the index goes down, the major can grow because total variance is growing, and it allows the major to grow. So the maximum you can see it is at the intersect of 50%. And then after that, what happens again? It goes down. Why does it go down when total variance is going up? Because it's now it's restricted by the index definition. Index, because it cannot be bigger than the index. So that's fine. I mean, the pyramid shape is fine, but it's what part of the pyramid does it occupy? You can see a lot of it is at the early part and very few at the later part, and some of them do reach the peak. And for the human, it's not exactly the same as the human. So we see, so as the avian, so we see very similar, but there are some differences too. And among the avian, you can see that they're coming you do see some of them competing with the index. They're coming very close to the index, not so much. Well, there are also some there, but not so much in the human. So that's one of the things that we could see. Then the next one was the minor and the unique. Actually, I've gotten this a lot. A lot of people would think the unique could be mistakes, sequencing error, since you only see them once. But what happens is actually as a population, we see about like 5 to 10% of them. That's quite high, for it to be sequencing error. And so this unique, you see, quite a good distribution of them. In fact, for HIV, you see a lot more. So we don't think this is sequencing error. This actually not artifacts, but they seem to be happening. Well, some of them could be artifacts, but as a pool, this seems to be something that's out there. All right, so the minor seems to be quite a dominant. Minor is the one that exceeds, goes beyond the index. As a population, they can go beyond. So the minor seems to be a big challenge here. The major can be a challenge to the index. The minor as a pool can be a big challenge, and the unique, are interesting aspects by themselves. But they're only found in certain locations. They're found actually in majority of the locations, but big proportion of unique is in certain locations. And of course, you see a lot of those when total variance goes up. Now let's just see for the nonotypes. So we do see very small nonotypes, but about 10% out there. But you see, interestingly, you see more nonotypes for humans. So this is also a concern that for human, although the entropy was rather similar, total variance was rather similar, but you see a lot more nonotypes, a lot more variants coming up for the human viruses. So this is something, you know, for vaccine work, they need to be wary of this and actually look into this further. Let's compare with the HIV. So it's very interesting. For HIV, index is almost a straight line all the way from 100% to actually, I think the lowest index was 2%, index of 2%. That's just amazing, which means your total variance was 98%. So imagine when you have total variance of 98%, index is 2%, your major variant has to be below 2% by definition. Your minor has to be below 2%, your unique has to be below 2%. So is it possible as a total variance, they're 98%, but everything has to be below 2%? The only way you can do that is if you have a lot of these variants. So at those positions, there were just a lot. And you can see the unique variants, they're just curving up. And so these are, we used to call this, we call them hyper variable positions, hotspots where mutations or changes are just so rampant, almost the plastic definition of an HIV virus can be seen at those positions. Not so many of these positions, but there are such positions in HIV. So it's almost a straight line all the way, total variance almost a straight line. And you can see the major variance is rather different. It's quite well distributed, unlike the flu. So we learned something about the viruses by doing this kind of a plot. And the nonotypes, yes, much higher, close to 60% at some of these positions, especially at those positions where your index total variance was like 98%. And so of course, we did the same thing for the individual proteins, and the proteome was a collective thing. The individual proteins, if there's an interest, you can look at HA, NA, for example, and see what's going on. And I don't have time for that, but we did that too. We have all that data, and you can see for some of these proteins, the index just stops at like 60% or 40. Somewhere in the middle itself, so you can see that diversity is rather limited for some of these proteins, but HA goes all the way. NA is also quite bad, so a lot of that at the proteome was probably a reflection of what's going on at the HA level and the NA level. So you've got to also look at the proteins just to understand what part of the proteome is contributed by the different proteins. So this was useful. This was some sort of a Violin plot that was quite helpful. It gives you a very good summary of what's going on. So the index, of course, you have a lot, big bulge there at 100%. So a lot of them were at 100%, but very small proportion, had 25% incidence of the index. And of course, the inverse would be, and we don't have a plot for total variance here, but so the inverse will be for total variance, but we've broken down total variance into majors. You can see the major variant, a lot of them are very low, but there's some that actually, come pretty close to 50%. Minor variants are rather high, unique is there, and nonotypes. And you can see the major variant of the human is not exactly very similar to the major variant of the avian. There's a different kind of a pattern there. So this again can be studied further. We don't have time to go into the details, but it gives you an idea, the uniqueness of, just same subtype H5N1, avian and human, you can see some differences there. And this is to compare with HIV. Just look at the index of HIV. Every part of that spectrum is covered. The major variant is quite different. The minor is such a huge bulge up all the way up there to 80%. The unique variants are very different, and nonotypes, So, and this gives you a perspective that, what we can learn from these different viruses. And of course, we do the same for the individual proteins. We get to appreciate the detail, the devil is always in the details. So you got to look at these contributing proteins. Okay, so I want to quickly, change focus. I'm so sorry I've taken so much time very quickly. There's some interesting concept called index switching. And this is usually happens at these positions. So you saw for HIV, we have a lot of these major variants that are very close to the index. So what happens is the idea of a switch, the major variant actually switches and becomes the index, and the index becomes major variants. We observed this the first time with HIV, and we took it further for flu. So we observed index switching, and we were wondering whether motif switching actually happens. So this is to demonstrate, for example, at this position 2349, position 168, 2,300 sequences, 300 plus sequences, and you see the index is this one, and these are your variants, and you have 67 more variants. but these are the key ones. And then when you go to position 169, the index still remains the same, but there's some changes. Your major variant is now 491 because as you moved one position, some of these sequences get merged together. There'll be some splits, some merges. So you see 491, 489. When you move one position, this Q disappeared. So these two basically got absorbed by the major. So it became 489 plus 2, you get 491. So that's for the major. But I think let me highlight the index switching first. But you notice here at the position 170, NST now has become the index. So there's a switch here. KDNA, DNA was supposed to be the index, and it's supposed to be indexed all the way. You do these monomers and overlapping monomer positions supposed to be all the way, but there's a switch in this position. The DNA became the major and the index is taken over. What happens is in the next position, it comes back. The major now comes back to become the original index comes back. All right, so we observed this, and this was quite interesting. And we also highlighted that actually this is not restricted to just the index. It doesn't happen only for the index. It actually happens between the various motifs. So it's quite messier than what we had thought. So this plot kind of demonstrates the mess. You can read more about this in the paper. So all kinds of switching actually happens, and this is something that we suspect could actually contribute to this immune escape. You target you could be targeting one sequence, and the immune system recognizes, and there are positions where the switch happens, and that could affect the immune recognition. We discussed this further in the paper. You could read more about it, but we think this has important implications in terms of vaccine design or immune recognition. And this is one of the ways we suspect, the virus actually utilizes or gets, benefits the virus in terms of evading recognition. And of course, subsequently from the data, you can do, you can break it down into highly conserved, highly diverse, and something in the middle, mixed variable. And then we can have a criteria. Highly conserved is like 90%, index of 90% or more. Highly diverse index below 20% or below is highly diverse, and something in between. You could change this depending on your virus of interest. And then you can have, this distribution plots, and you can then extract out these highly conserved sequences, and then you have all kinds of application. You can do vaccine design, drug design, or even diagnostic markers. So this is, something else that is a byproduct of that analysis. So I mentioned WADA. So, we're trying to automate this so other people can do this. In conclusion, I just wanted to, since I'm already overshooting. And so what did we learn? So the first key message is that there's a large repertoire of viral variants. We think of, I don't know. So for example, there are viruses and their variants. We know that, but what was amazing, what is interesting is that at every one of these overlapping positions, you see a lot of these variants. So there's a big repertoire of variants, and it's most amazing for HIV to see how actually that many variants can happen. You can actually have index of 2% and total variance of 98%. We didn't see that for flu, but yeah, there are a lot of variants. I think for HIV, every position, I think there were no completely conserved. So every position there were variants, or at least majority of the positions in flu, there were variants. So you expect, so you can't simply pick a position for vaccine design. I think we got to be careful with throwing a whole virus, and when we throw a whole virus for vaccine, we don't really know what gets recognized for vaccine, by the immune system and presented and goes into memory. So we have to have much more targeted approach because the variants are everywhere. So this calls for such a targeted approach, and the motifs. We feel that the motifs highlight some aspects of the diversity. They allow us. It's a tool that allows us to better understand the diversity. And we actually saw distinctive pattern, which hopefully most likely represent, intrinsic pattern within the structure function organization of the viruses that depict the fitness selection of the quasi species that we, for those viral populations. And we also observe this complex motif switching, and this has implication to fitness selection of certain strains, and of course further implication in terms of immune recognition, vaccine design. And despite all this, we still see that there are highly conserved sequences, even in HIV, despite it being so plastic with all kinds of mutation. We still saw positions that were quite conserved. Maybe not completely conserved, or you have very few completely conserved, but you see a lot of you do see a good proportion of highly conserved sequences, even in HIV. So that was the good news, and these groups might be the way to beat in terms of vaccine design, and that's what we might want to focus on. But we can't just randomly. We definitely have to have targeted approach. We can identify those positions and then further investigate them as vaccine targets. All right, so I've come to the end. I just want to acknowledge a number of people from Perdana University, Malaysia, Hadia, who actually did the H5N1 work as a part of her MSc. Tan Swan actually looked at H7N9, which I did not discuss, but she was one of the early ones that contributed into the development of this work. Of course, our student Lee Chun Chong, who did the alignment independent. I didn't cover much about her work, but she is looking at the other aspect. Hopefully we'll get to publish that, and we will have two different ways of looking at diversity. Mohammed Farhan Shaugi is actually looking at the development of the tool, the WADA tool. So he's working hard on that. And from National University of Singapore, we have my mentors. One of them is Prof. Tan Tin Wee, who has been very instrumental and very supportive of this work, and Dr. Hu Yong Lee, who actually did the HIV work as part of her PhD. So a big thank you there. And Dr. Olivo Mioto, who actually contributed the ABK Awana platform, which was one of the starting points for the work in this direction. My other mentor, late Professor Thomas August, who passed away sometime recently. So we miss him dearly, and we thank him for all the things he did for the project. And my other mentor, Prof. Vladimir Bruzik, who is currently based at University of Nottingham, Ningbo, China. And he has been also a big starting point for this work. In fact, the starter for all this that I have presented here. Funding, we thank NIH USA for the earlier grants to late Prof. August and Prof. Vladimir Bruzik, which kind of started the work. Perdana University for the Faculty Development Fund. Bezmi Ali Wakif University for my support being here in Turkey, and of course the Scientific and Technological Research Council of Turkey for the International Fellowship for Outstanding Researchers Program 2232 that they have awarded to me. So all in all, thank you very much. I hope that was useful. I hope you all learned something and some take-home messages, and I hope more of you would explore research in research of viruses. Thank you so much. Abhishek, back to you.
Thank you, Dr. Asif. Very nice talk, and we are very, very thrilled to see that you are working with the viruses and yeah, the variation by taking short peptides. So something on similar pattern we are working, but not thought in this way, but will worth seeing. I just saw your PJ paper some days back, and we also have devised some of the motifs from SARS-CoV-2. And we think that there can be good candidates. So basically, in our future study, and hopefully some of my students are seeing that we can employ your methods.
Sure.
And so
we'll be happy to help. Yeah,
stage is open for questions. So let's see if they have questions. Yeah.
Okay.
So I just check it in YouTube and also here. Okay, let me have some time. I feel still a lot to learn from Zoom. So let me go to the YouTube and okay, so live. Still a lot to learn from Zoom. So
you can also email me if there are any questions.
So basically
my emails are here. Yeah.
So is my question is there how, say, in coronavirus, do you see a common stretch or coronavirus, or philosophically just assume that any virus, yeah, we,
Mhm.
where you can have what device one uh, uh, small peptide to target entire family of viruses, like say.
Yeah, one peptide to target entire family of viruses. For example, for the H5N1 work, I actually skipped some slides I could show you. So yeah, so for example, we actually found 12 peptides that were pan subtypes, so they could cover a number of a number of flu subtypes, at least the key ones. So there were 12 of these. Of course, as you start covering more and more, and these are subtypes of one species group. If you start going to the various families, it depends on the overall diversity of that family. If the diversity is not much, you could cover more species under the family. If the diversity is a lot, you're probably gonna be finding it difficult to do that. And with influenza, the diversity is there, but we managed to find at least 12. No, actually 8 that were actually pan subtype and immunogenic, so much smaller. So not only they have to be pan subtype, they also have to be immunogenic. And immunogenicity is something else that I didn't discuss. That's another area. But yeah, the number will get smaller. You could find something that's pan subtype, but one for whole family, it's gonna be a difficult task. Not a difficult task. It's gonna be difficult to find. Um, but it depends on the diversity of that virus. It might be possible, but it's going to be challenge. And people have been looking for this universal vaccine for flu. So there is hope, because we do see some of these segments or parts that are highly conserved across the board. But this is not completely conserved. We must bear in mind they are not 100%
Yes,
conserved in all.
my idea
So
is
just by
that
80%, yeah.
can
So
we
if
have a
we lower our threshold,
peptide?
maybe we can. Sorry, go
Can we
ahead.
have a cocktail of 10 peptides, something like this?
Ah,
Because
yes, yes. So you
uh,
could do that. Yeah, you could do that with a cocktail approach. Maybe one covers 50%, another covers the other 30%, and so on. So you could do a cocktail peptide approach. However, there's a problem with cocktail approach. For example, in dengue, they have observed something called I think immune interference. So when you have a cocktail, you got to be. Well, this is I guess an additional work that needs to be studied, whether putting all of them together would have some sort of a side effect in terms of the recognition by the immune system. How would that play out in terms of recognition? Or should you do it in sequential, with one peptide and then the next one, the next one, the next one? I think this is something that is being explored and discussed. I'm not sure there's a perfect formula for that yet. So yeah, immune interference, it's something out there that needs to be thought about. But the idea is, yes, we could cover the diversity through a cocktail, but how would that cocktail work out? The other thing is the diversity of the immune system. Your HLA, you have such a huge diversity of HLA, and you need to look into those promiscuous epitopes that would give you a good coverage of the immune system in the population, and that way. But yeah, in general, you're right. That's something that could be a direction forward. But overall, I don't see a lot of people actually focusing on the. I mean, there is effort, but compared to the traditional approach, the other approach of peptide-based vaccine or epitope-based vaccine, this is happening, but the focus is not so much compared to the other side. So hopefully, more and more people will do more research on this. This side has its own challenges, but hopefully, we would overcome those challenges.
yeah, so basically it's a very interesting talk, and we have also submitted on one paper with it more than one peptide approach, small peptide, to suggestion that these can be future vaccine targets, and especially
Congrats.
peptide based recombinant peptide based approach, and but we do have not tested it actually, so this is now under review in,
Mm.
and we got some comment to work with, but then
Right.
basically with SARS-CoV we have started working with viruses, so in the near future we'll have a
All right.
good cooperation. So our focus is not just to resolve SARS-CoV, like not very quickly get published, but to establish its opportunity for us to establish a little bit into viruses. First, we have to understand it, because our virus is limited. My virus knowledge was limited before to SARS-CoV. Yes, so I never focused on it, primarily because of other constraint that I was working in different areas. And what we are now doing is that we have a focused team working and reading a lot about viruses. And now so in near future, maybe we will present our work to you, and then see how what is your opinion about it, because you are working with viruses. With this, I would like to thank you again and for your valuable time, and I'm extremely sorry for that time constraint that day and time. As I told you, there were two person, and both you and the other person I was talking, who can actually shift it one after. So there was a kind of a, this is happening in online and time zone difference, due to time zone difference. So we'll learn from it, and we'll try to get it. Next day we were on exactly on the time. We do not allow any speaker to stretch. So yeah, it was learning for us, and thank you that you are agreeing, and that with this our conference is officially ending. And
congratulations.
So like, until unless you publish your paper, it's not published, even if it is a minor comment left. It's one special talk was left. With this, we are coming to end. And for the international conference in data science in biology, would like some two other groups to take on, maybe as a virtual conference itself, for next year. So if someone is interested, it's open to for discussion. And so there are two other groups, or one of us and some other another person, if they want to take over, it's open to discuss actually. So
it was a pleasure being here, and I again congratulate you and the team for the wonderful work. And I think you had very little time to prepare for all this, so I've seen how you've done it so professionally and put in a lot of effort. I think it's a challenge for all of us to figure out what's the best platform and the best way to get these such virtual conferences out there. We have our very own INCOP coming out in November, so we'll be learning from you. Hopefully, we
have a backend team we are hosting with support of eDot and Cassid. They are two software companies, small software companies actually, and they provide that. So we do not have our. So I'm able to talk to you properly, and they are managing that. If I got unmuted, or they are taking care of the minor hitches, and then they are editing all the videos. So your live videos is going now live, but edited video will come to you at the end of maybe end of the week or so. And so they are taking care of those areas. So we are lucky to have them. And plus we have the BioClues organization, other organizations. BioClues we have taken members from, and from BioClues only I got connected to you. So or Apo BioNet, you have two members, you and madam, yeah. So
Prof. Shobha, yes,
Professor Shobha, madam. And so that's societal benefits we get. And then these are the supporting support we receive. So basically one has to look for the support.
Agree.
So with this we come up to an end, and we again thank you everyone.
Thank you very much. Okay.
So so with this we end up. So have a nice time. Thank you very much, and thanks for your valuable time out of the.
All right. Thank you so much, and it was a pleasure. We learned from each other, and all the best, and hopefully more of such conferences in the future. Okay.
Yeah.
Cheers.
Thank you.
Okay. Bye-bye.
Okay. Hello.